Good evening. Uh, nobody has joined yet for the live session, but I'll get started anyways, and people can then watch the this since this is being recorded. Uh, People can then watch the video later on. So the format of the live session remains the same as before. I'll go through a set of MCQs, and then I'll discuss each of the MCQs and the concepts related to those MCQs. So let's get started. So we are. This is the 11th live interaction session, and we are discussing week seven's content right now, which is the motor cortex. We got started with the motor cortex last week, and we discussed one question which was related to Graziano's work. So we'll now continue. Uh, just trying to see how to ink this. So the tracts from the following region of the brain do not connect directly to the spinal cord or the brain's stem. Uh, so let us see what tracts are. Basically a set of neurons that are traveling from one region of from the brain to another region of the brain or maybe the spinal cord. So that is a tract, which do not connect directly to the spinal cord or the brainstem. So we know that the primary motor cortex and the uh, premotor areas, that is the dorsal premotor area and the ventral premotor area, directly have tracts that directly go to the spinal cord. So there are corticomotor neurons in these areas. So let us recall what corticomotor neurons are. Corticomotor neurons are those neurons which travel from directly from the cortex to the spinal cord. So these are the which corticospinal neurons. So these are the corticospinal neurons, and those neurons that from the cortex that directly synapse onto the alpha motor neurons, which control the muscles, are called corticomotor neurons. So, however, in the pre-supplementary motor area, the the neurons or the tracts don't directly go to the spinal cord or the brainstem, but they indirectly go to the spinal cord and the brainstem through different cortical areas, through projections to different cortical areas, and uh, and also through different subcortical areas. So the answer is B, pre-supplementary motor area. Just discuss the next question. The tracts from the cortical neurons that go only to the brainstem are called as A. Corticospinal tract, B. Corticobulbar tract, C. Rubrospinal tract, or D. Olivospinal tract. I think somebody has joined. No, nobody has joined. Stay in the call. So you can pause the video at this particular location and try and answer the question yourself, and then we'll discuss this question. So, so the tracts that go from the cortex to the spine are called as corticospinal. Is called as the corticospinal tract, and the tract that goes from the cortex to the brainstem is actually called the corticobulbar tract. So this is the correct answer. Answer is B. So we know that these two tracts together form the what is called as the pyramidal tract, and there are other areas which are the subcortical areas in the brain, which also project to the spinal cord. For example, this is the rubrospinal tract. Uh, so these are projections from a subcortical area called the red nucleus to the spinal cord, and olivospinal tract is again a subcortical area, where olive is a subcortical area, and projection from the olives to the spine to the spinal cord is called as the olivospinal tract. And then there are other such tracts, such as from which project from the subcortical area to the spinal cord, such as the rubrospinal tract, which projects from the red nucleus. Uh, Nobody has joined. And then we have the reticulospinal tract, which projects from the reticular formation. These are all subcortical structures. So, corticospinal tract plus corticobulbar tract is called as the pyramid. This is called as the pyramidal tract together. And projections from subcortical areas to spinal cord. These are called as the extrapyramidal tracts. So let me just show you how it looks. We have seen this in the slides, but let's just go through it. So this is the situation. So we have the areas in the red are the descending pathways. So this is a transection of the spinal cord, and this is the anterior region, and this is the posterior region. So we can see that the pyramidal tract consists of the corticospinal tracts, and the extrapyramidal tract consists of the rubrospinal tract, reticulospinal tract, olivospinal tract, and vestibulospinal tract. And we can also see that here there is no corticobulbar tract, because uh, this uh, this is uh, this is a transection of the spinal cord. So, but we know that the corticobulbar tract starts at the cortex and ends at the brainstem. Okay, so the observed effects of lesions depends upon location of lesion, species in which lesion is observed, size of the lesion, or all of the above. So again, you can just pause the video and try and answer the question yourself. So we know that the effect of lesion is dependent on all these three factors. For example, where uh, where the lesion is located determines which functionality is lost. For example, if we cause a lesion in the motor cortex, in the primary motor cortex, then a particular kind of functionality will be lost. If we cause a lesion in the premotor cortex, for example, in the dorsal premotor cortex, if there's a lesion, or if there's a lesion in the ventral premotor cortex, both of these lesions will cause different effects. Similarly, in the supplementary motor area, if there's a lesion, then that leads to other kinds of effects. So this has all been experimentally shown. So it is easy for us to jump to the conclusion that uh, the lesioned area. So if we lesion an area and a particular function is lost, for example, fine control of movements is lost. If we let us say, we cause a big lesion in the motor cortex, that leads to paralysis in the body. So it is easy to say that the motor cortex controls that, and the entire functionality of movement is controlled completely by the motor cortex. Or for example, if we cause a lesion in dorsal premotor cortex, then uh, or let's say ventral premotor cortex, then what happens is uh, the ability to pre-shape the hand. So if a person is going to grasp an object, then what happens is, uh, based on the size of the object and its texture, we pre-shape our hand so that we can grasp the object. But it has been shown that lesions in that area uh, completely eliminates the ability to pre-shape the hand. Uh, so we are not able to process the visual input and then use that visual input to give the appropriate motor commands. So it is tempting to say that the this particular function is localized in that area, but we cannot make that claim because this particular area can be a part of a long neural circuit, and only just and it disrupts this neural circuit. So we cannot say that that particular function is localized in that region alone. Okay. Then the next thing is species in which the lesion is observed. So we know that, for example, lesions of the upper motor neurons in lower mammals, uh, or for example in rats, is less devastating when compared to the same lesion caused in higher mammals, for example in monkeys or in humans. So if we lesion the upper motor neuron in humans, we know that there are direct monosynaptic connections from the motor cortex to the alpha motor neurons that control the movement. So if this is destroyed, if there is a lesion, then basically the control is there is no control. So the effect is more devastating. But in the case of lower mammals, we know that the number of monosynaptic connections are less. So since these monosynaptic connections are less, the signal goes through different, for example, it projects onto interneurons, and then there is processing happening there, and then the motor command goes to the alpha motor neuron. So if the upper motor neuron is damaged, then there are still ways through which recovery can happen, and the command can the brain or the brain, the lesioned area can still reorganize itself to accommodate for this perturbation or this lesion. But in the case of humans, there is no way to accommodate for this because that is the only pathway. So if that single pathway is destroyed, then it's going to be very hard for recovery to take place. So, however, the advantage of having the that as the only path, the monosynaptic projections, is that we get fine dexterity and control. So that is the advantage. But then it also comes with the disadvantage. So we see that in humans the fine control of we have there is more fine control of the hands and fingers when compared to other animals lower down the evolutionary tree. So species in which the lesion is observed also matters, and then of course the size of the lesion. For example, if we do a focal lesion in the primary motor cortex, the effect is going to be something, and if we do a large lesion in the primary motor cortex, the effect is going to be different. Uh, so all these three factors come into the picture. So the answer is D, all of the above. Okay. Regular practice of motor skills prevents functional degradation of the brain area responsible for the skill, structural degradation of the brain area responsible for the skill, both functional and structural degradation of the brain area responsible for the skill, and D, none of the above. So, one of these questions we can answer directly by our own experience. For example, if we are practicing a particular skill, let's say a guitar or playing the tabla, and if we are practicing it on a regular basis, there is no functional degradation of the skill, right? But if we stop practicing that particular instrument, then let's say for 2 months, and then if we come back and we try to play the instrument, it won't be the same as how it was 2 months ago, when we were regularly practicing. So there is a functional degradation, and to get back to that level, we again need to practice for some time to reach the original level at which we were before we stopped playing. So then that is called functional degradation. So this is obvious. And then we have structural degradation of the brain area. So what this means is, uh, so it has been experimentally shown that practicing a motor skill causes neural plasticity in the brain. Uh, so this work has been was first pioneered in 1996 by Mike Merznick, where he showed that uh, before practicing a particular skill, the area that was represented by that skill, the movements that were required to perform that particular skill uh, in the brain was less. So the representation of that skill in the brain in terms of the area was less. But post practice, after regular practice, it was noticed that the area in the brain that was associated with that skill expanded. Uh, so this was a structural change that happened due to practice. So there is neural plasticity in the brain. The organization, the functional organization, as well as the anatomical organization of the brain is a dynamic process. Uh, so, so with practice, it was shown that uh, there was structural changes in the brain. Uh, so there is both functional, and so if you don't practice, again that with function, as the function is also lost, there's also structural degradation of the brain area responsible for the skill. So the answer is C. Okay, so match the following. I'll just go out and fill some water. In the meantime, you can try and answer this question. Okay. Okay, so let us discuss this particular question. Uh, so match the following: Lawrence and Corpus. So what he did work related to lesion studies. So pre-lesion and post-lesion. Uh, what happens? Let us see what he did. Uh, what he did was he investigated lesions in the pyramidal tract, and what he found was that lesions in the pyramidal tract abolish fine grasping movements. So let me just go to the notes and just show you. So this was done in way back in 1968. So what he did was he lesioned the pyramidal tract, and before the lesion, the monkey had to grasp a particular food item or a treat in a small well kind of setting. So you can see here that there's this small well, and within the well there's a food item. So to pick up the food item requires fine movements of the thumb and the fingers. Uh, so post lesion of the pyramidal tract, what happens is it was noticed that the movements were claw-like, and the fine movements were the monkey was not able to make the fine movements. So this is what he did. So 1B, then to Mark Shieber. We have seen what Mark Shieber has done. Uh, if you recall from the videos, uh, what he showed was that the somatotopy in the motor cortex hand area is not that much. So what do we mean by somatotopy? By somatotopy, we mean that one particular area in the brain controls a particular area in the body. So, for example, if I stimulate a particular area in the brain, uh, then that particular area complete should lead to contraction of a particular area in the body. However, what he showed was as we zoom in to the motor cortex hand area. Uh, so, for example, if you consider the fingers, there is the neurons that control the particular finger muscles are spread across in the hand area. They're not localized to one particular area, but rather they are uh, spread across the hand motor cortex area. So that is what he showed, and he also observed that some neurons in the cortex area also controlled multiple muscles. So it is not that there is a one-to-one mapping between the neuron and the muscle that it controls. So that is what Mark Shieber did. So that's 2B, 2A. It should be 2A. Then what Busy did was he did work related to adaptation of force fields. So he conducted experiments where there was reaching tasks and a force field was applied, and he investigated the nature of learning. So that is 3D. And then we have Carl Wernicke. So we know that areas related to speech and language are localized in the one particular hemisphere. That's 4C. So let us see what the option is: 4C, 3D, 2A, and 1B. Answer is C. Another match the following question. Uh, again, what you can do is you can try and answer this question, and then we'll have a discussion. And then I'll discuss this MCQ. So, lesions in the dorsal premotor cortex. Lesions in the dorsal premotor cortex. Uh, then lesions in the ventral premotor cortex. Lesions in the motor cortex, and then we have pyramidal tract lesions. So let us just write it down here. 1, 2, 3, and 4. So let us first see lesion in the motor cortex. So we know that since the motor cortex directly sends the commands for movement, large lesions in the motor cortex can lead to a complete abolition of movement. So it can lead to complete paralysis. So 3B. Then we have pyramidal tract lesions in monkeys. So this leads to uh, pyramidal tract lesions we have seen leads to clumsy claw-like grip. So we have seen that there are many symptoms when there's a lesion to the pyramidal cortex, when there's a lesion to the pyramidal tract. One of those symptoms, so those symptoms together are called as a pyramidal syndrome, and one of those uh, symptoms is clumsy claw-like grip. So that is, we have seen that in children. What happens is when the movement of the when the control of the hand movements is not properly developed, we see that there's flexion extension of all the fingers together, but there's no individual control of the fingers. Uh, so that something similar is observed. So 4C. Then in the ventral premotor cortex, I just explained some time back, the ability to pre-shape based on the visual information is lost. So that is 2A. And then we have the lesions in the dorsal premotor cortex. So what happens in the dorsal premotor cortex is that when the lesion happens, the ability to map the sensory information to the motor command is lost. So this the learned association is lost. So and yeah, so the answer is 1D. 1D, 2A, 3B, and 4C. Answer should be B. Okay, moving on to the next question. Which of the following statements is false with regard to Georgopoulos' work? Again, you can uh, just pause the video and try and answer the question yourself. So we know that. Uh, let us look at what Georgopoulos did. Georgopoulos was the one who first championed this center out reaching task, which is a very popular paradigm, which became a very popular paradigm and led to a lot of different experiments. So he was the first one to do this. So what he did was a monkey had to there. This was a home position, and the monkey had to reach in a particular direction to a target. And what they observed was, so this is the center of teaching task, and what they did was they collected the recordings from a whole bunch of neurons, from a population of neurons, for example 180 to 200 or maybe more, uh, and then they tried to analyze the data, and they came up with a certain conclusion based on the observations. So this is the neuronal activity of a single neuron during the center out reaching task. So this is called a raster plot. So each row here in you can see that there are different rows here. So each row shows the neuronal activity of a single neuron. So for example, here, so when the monkey is reaching from the center towards this direction, so these are 5 trials of data here that is shown. So this is the neuronal activity, and we can see that 0 marks the onset of movement. So slightly before the onset of movement, we can see that there's high level of neuronal activity. So while reaching that towards the 180-degree direction, we can see that the neuronal activity is high. Similarly, while reaching in the 135-degree direction, the neuronal activity is very high. And you can see that progressively in the so in this particular direction, the neuronal activity is high, but in the opposite direction, we can see that the neuron is completely silent. So there is a preferred direction for each of they did this for all neurons, and they found that there's a preferred direction during which a particular neuron fires very in a has maximum firing. So that preferred direction of that neuron. However, there is no there's a gradation there, which is called a directional tuning curve. So we can see here that in this particular direction, the neuronal activity is high, and in as the direction changes, the neuronal activity progressively decreases, and then finally it becomes 0 in the opposite direction. It's completely silent. So this is the directional tuning curve. Uh, so what they did was uh, they found out that so in this particular plot, what they did was for example, let us consider this case: the monkey is reaching from 0 from the home position to 0 degrees. Uh, this is a population. They collected the population, the neuronal activity from the entire population of neurons, and here for each neuron they created a vector, and where the direction of the vector signifies the preferred direction of that particular neuron. So we know that, for example, in this neuron, the preferred direction is 180 degrees. So here they first the vector of a particular neuron represents its preferred direction, and its magnitude represents its neuronal activity during that particular moment. So we can see here. Uh, so for all the neurons, they created for the entire population of neurons, they created this particular vector, and then they finally did a resultant. They came up with a resultant vector. So they added all these vectors up, and they came up with the resultant vector. And what they found was that during this population of neurons can actually predict the neuronal activity from the neuronal activity. The direction of movement can be predicted. We can see while reaching here, the direction of the resultant vector is pointing in this direction, which is derived from the population of neurons. Similarly, for all these directions, we can see that the direction of movement and the direction of the population vector is approximately the same. So what does this say? This says that uh, the movement kinematics, or the direction of movement, is coded in the population of neurons. Uh, so that was the conclusion. This was uh, the first of its kind. Uh, so let us go back to this particular question. Which of the following statements is false with regard to Giorgio Polis's work? Neuronal activity of a single neuron encodes direction of muscle. No, it encodes direction of movement. So this is wrong. Neuronal activity for population of neurons encodes direction of movement. This is correct. All neurons are highly active and moving in a particular direction. No, each neuron is active in its preferred direction. So it's active only when the movement is in the preferred direction. And different neurons have different preferred direction. That is correct. So the answer is B and D. Which of the following statements is false? Humans have less direct monosynaptic projections. Let me just drink some water. Humans have less direct monosynaptic projections from cortical neurons. Let me just go to the previous one. This is question 8. So the problem here is we have to see which is false, right? So these two are false. A and C are false. So the answer should be A and C. A and C. Okay. Going to the next question. Which of the following statements are false? So again, we should see which of the statements are false. So first we'll see which of the statements are correct and false, and then finally we will see, write down the answer. Humans have less direct monosynaptic projections from cortical neurons to distal parts of the hand compared to monkeys. This is false. In humans, the number of monosynaptic projections to the distal parts of the hand are the highest, and the number of monosynaptic projections progressively decreases as we go down the evolutionary tree. And this is the reason why humans have fine control of the hands, and the dexterity of the human hand is unparalleled when compared to other animals. Uh, B. It is possible that sometimes focal lesions induce devastating effects and large lesions produce lesser effects. Yes, this is true. Sometimes large amount of lesions can lead to, it again depends on the location of the lesion, can lead to less visible defects, and focal lesions can lead to more devastating effects. But this is not a generic statement. It depends on where the lesion is, uh, and what is the size of the lesion. So this is a correct statement. So again, the important word here is sometimes. Humans recover much quickly from a lesion compared to cats and monkeys. This is false. Cats and monkeys recover faster than humans, and that's obvious because the situation is more complex in humans, and especially with the monosynaptic projections. If these monosynaptic projections are lesioned, then it's very hard for recovery to take place. And deep pyramidal tract lesions abolishes deep reflexes while heightening superficial reflexes. This is wrong. It's the opposite that happens. It's in the pyramidal tract lesions, the deep reflexes are heightened and the superficial reflexes are abolished. So the answer is, which are false? The answer is A, C, and D. In smaller mammals like rats, post upper neuronal regions, significant time is required for recovery. This is not true. Reorganization happens where adjacent areas take over, but after considerable time. This is also not true. Reorganization happens where adjacent areas take over within a short duration of time. This is true. And reorganization depends on the horizontal projections between the two adjacent areas and local inhibitory circuits. Yes, this is true. So we have seen this. Let us just open up that slide. So in the rat, this is a work done by Donahue. What they saw was uh, so this green area is the area which controls the upper lip or the whiskers. So, for example, stimulating in this is the situation in a rat. So, when this area is stimulated, then what happens is the whiskers or the upper lip gets of the rat gets stimulated. So, this is the area that controls that. But when the nerve that controls uh, this particular area is cut, what happens is within a few minutes, not even hours, within a few minutes, the adjacent areas, especially the forelimb area, takes over the whiskers. So now, stimulating the same area will lead to movements in the forelimb of the rat. And because the connections there are Uh, Let me go back to that slide. The correct word is horizontal projection. The horizontal projections are more between the forelimb and the whisker area. Uh, this plays a part in this area being taken over by the forelimb area, and lesser by the other areas. So the answer is C and D. Okay. Going on to the next question. Select the statements that are false. Upper motor neuronal lesion causes denervation atrophy. Uh, this is actually a false statement. This is because lower motor neuronal lesions causes denervation atrophy, but upper because essentially what happens is the alpha motor neuron in the lower motor neuron in the upper motor neuron, the nerve is intact. The alpha motor neuron is still intact, but it is not receiving signals from the upper motor neurons. So what happens is uh, there is disuse of the muscles, and then this causes disuse atrophy. So upper motor neuronal lesion causes disuse atrophy, whereas lower motor neuronal lesion causes denervation atrophy, because the nerve itself is in the alpha motor. If the alpha motor neuron is destroyed, which is the case in the lower motor neuron lesion, uh, there is no nerve, or there is no neuron that is supplying the muscle. So in that case, it leads to denervation atrophy. So as the saying goes, you either lose it or you use it, or you lose it. So if these muscles are not being used, it leads to the atrophy of the muscles. A Babinski reflex is observed in lower motor neuronal lesions. This is false. It's observed in upper motor neuronal lesions. So let us recall what the Babinski reflex is. So in children, if you take a blunt object and you run it along the foot of the child, what happens is the thumb extends, and there is fanning of the, and all the toes of the leg extend, and there's fanning of the the remaining toes. So that is the Babinski sign, or the Babinski reflex. And in adults, what happens is this Babinski reflex is not there. When the same, if a blunt object is taken and it is lightly run over the uh, foot of an adult, what happens is there is flexion of the toes. But in the case of upper motor neuronal lesions, this same Babinski reflex is observed. Instead of flexion, the Babinski reflex is observed. Then upper motor neuronal lesion results in hypertonia, whereas lower motor neuronal lesions cause hypotonia. This is correct. Upper motor neuronal lesion results in spastic paralysis. This is correct. So there's a heightened tone, and there is a rate dependent rigidity also that is observed. Whereas lower motor neuronal lesions results in flaccid paralysis, or there is it's loose. The muscles are and limp. This is correct. So the answer is A and B, because these are the two false. We have to find the false statements. Okay. Going on to the next. Lesions in the pyramidal tract result in ipsilateral paralysis in most cases. This is false. It's contralateral paralysis. Hypotonia preceded by hypertonia. This is also false. It's the opposite. It should be hypertonia, which is preceded by hypotonia. Spasticity is correct. We just saw it in the previous this thing. Uh, and Babinski reflects in adults. The answer is C and D. So there is a heightened muscle tone because of loss of the descending inputs. That leads to which is causes spasticity or a heightened tone, muscle tone. So the answer is C and D. Okay. Which of the following statements regarding Peter Strick's experiments on corticomotor neurons are true? So, Peter Strick performed corticomotor motor neuronal recordings and observed that uh, the neurons that control arm muscles lie close to the anterior of the central sulcus. Let us just open up and see what this guy has done. Within the anterior bank of the central sulcus. This is Peter Strick. This is, I think, Peter Strick. It should be anterior bank of the central sulcus, but yeah, we can go for this. This answer is true. Perform neuronal stimulation of corticomotor neurons, that is correct, and observe that neurons from a given area diverge to different alpha motor neurons, but there is no convergence. This is false. Perform corticomotor neural recordings and observe that neurons from a given area converge to a single muscle, but there is no divergence. This is also false. Perform neuronal stimulation of corticomotor neurons and observe that there is both convergence and divergence from the neurons to the motor neuronal pool of different muscles. This is correct. Answer is A and B. So what he saw was, uh, by convergence, what we mean is, from a large number of neurons which is spread out in the in the area which the Peter Stick did this experiment. So it was the area controlling the motor neurons which supply a particular muscle or a particular motor neuronal pool are not confined to a particular area, but rather they came from uh, a large the it was spread across a large area in the motor map. And similarly, in some cases, what you saw was a particular from one particular area, a particular neuron controls multiple muscles. So there's both convergence and divergence. So that is what he did. Okay, this is a bigger question. Okay. Lesion of the alpha motor neurons in this, you have to select the correct statement. Lesion of the alpha motor neurons in the spinal cord is called as the upper motor neuron lesion. No, this is called lower motor neurons, not upper motor neurons. Lesion of the motor cortex above the level of the brainstem is called lower motor neuron lesion. Again, false. Twitching of an entire fiber or a group of fibers, muscle fibers, is called as fasciculation, whereas twitching of individual muscle fibers is called as fibrillation. This is correct, and this is noticed in the lower motor neuronal lesions. Neural plasticity is possible only at a younger age and not possible in older adults. No, neural plasticity is also possible in older adults, but it won't be as high as in younger age. But it is false to say that neural plasticity is not possible at all in older adults. Lesioning causes more severe effects in monkeys than in cats. This is correct. As we go up the evolutionary tree, the effect of lesions is more devastating. Twitching of an entire muscle or group of muscle fibers is called as fibrillation, whereas twitching of individual muscle fibers is called as fasciculation. This is false. It's the opposite. We already saw that in option C. In humans, lesioning a small area in the primary motor cortex causes paralysis, whereas lesioning a large area causes weakness and discoordination. This is false. It should be the opposite. Uh, Lesioning a large area should cause paralysis, whereas lesioning four lesions cause weakness and discoordination. And finally, the maximum effect of lesioning is seen in humans, especially in the hand and fingers, due to direct monosynaptic projection from the cortical neurons to the motor neurons of the hand and fingers. Yes, this is correct. So the answer is C, E, and H. C, E, and H. Okay, so there's one more question. Okay, we'll go through this question in the next class. For now, we'll stop the session here. Thank you and see you next week.